What's up, everybody? Welcome to USMLE Step 1 Preparation. My name is Tim. We're going to get started here. Today's lesson, the very first lecture in biochemistry, is about DNA and RNA, aka the nucleic acids. So we're going to be talking about the nucleic acids, how they're structured, and their organization, and how they're packaged as well. But we'll get into that as we go. So I'm going to be focusing in on more high yield, more talked about topics regarding DNA and RNA. So let's just get started with the central thing that we're supposed to know concerning DNA and RNA. And a lot of people refer to it as the central dogma of molecular biology. And I'm going to pull up a picture to show you guys as well. But basically you have DNA, which can then be transcribed to form RNA. And RNA can then be translated to form proteins. And proteins, as you guys already know, are the building blocks of all cellular function. And DNA contains the genetic codes to carry out the processes of life. So it's really important. Obviously, you guys know this already, but might as well mention it. So what is this called when DNA makes itself? When DNA replicates, it's called replication. And when DNA is transcribed, it's literally a code that's almost like typed and transcribed. It makes what? RNA. It's transcription. Wow, I can't even write. Transcription. Okay, now what happens when RNA gets made into proteins? That, of course, is when it becomes translated. So translation. And that's the central dogma. There is another step in there that we won't focus too much on, but it's important to note that if you reverse the transcription from RNA to DNA, it's called reverse transcription. Okay, so you guys got that? Good. On to the next thing. But this is the central... I really need to maybe fix this. Never mind that. So let's move on to gene expression and DNA replication. So basically, when cells divide, the daughter cells that form must receive the right copy of the genetic information from the parent cell. Next, we want to compare gene expression with DNA replication. And the difference is that when you're replicating DNA, it's making copies. But when you're expressing genes, that's when you're actually applying them, putting them into action. So basically, gene expression is what's going to produce the RNA and thus produce the proteins to carry out functions. And replication is just creating a copy and creating a copy for further generations, so it can be passed on. To figure out like the timing of how this happens in a cell, there's something called the cell cycle. And the cell cycle basically is a roadmap for the processes that the cell undergoes, whether it's replication, like we saw, division, let's take a look. So when we have the cell cycle, we can picture it as a big circle, right? Because it's a cycle. And like up here at the top, we have M, which stands for mitosis, right? You guys learned mitosis a long time ago, hopefully. And we won't really focus on mitosis, but that's basically normal cell division. I don't want to go too deep into it. And then there's a huge chunk over here, and that is called the G1 phase. G stands for growth. Other people say gap, but I think growth is more conducive to what's actually going on. So moving on next, after growth 1, G1, we have the S phase. And the S phase is when replication is occurring, when DNA is being synthesized, when it's being remade is the S phase. So G2 is the next phase, and this is when the transcription is occurring. So G2, you got to remember, is transcription. So G1 phase comes right before DNA synthesis, and it's basically the cell getting ready to synthesize more DNA, right? So a lot of cells are in G1 phase most of the time, and these are cells like epithelial cells and follicles, hair follicle cells, and also nucleated red cells. These are things that are in G1 phase. Now there's another phase that I didn't mention yet, and it's on the outside of the cell cycle because it's like a dormant phase, and it's called G0. Now cells in G0, nerve cells, muscle cells, these are in a resting state. So their DNA is just in a filing cabinet. It's not being replicated. They're resting cells. Most cells in the body are in a resting state. We have millions of neurons. We have millions of muscle cells. So G0 is known as the resting state. So cells can go from G1 back to G0, or G0 to G1. That can happen. And basically the cells, after DNA is synthesized, will go to G2, which is the after DNA synthesis, and replicated DNA in G2 is checked for any errors in transcription. So it's in a very important phase as well. And these errors in transcription is what's going to be the cause of mutations and thus the cause of many diseases. So next we really want to consider, what's going on in this phase? What's going on right here? Are genes being expressed in this phase here? The answer is no. No gene expression occurs during mitosis. And in G0 phase, does replication occur? No. Replication does not occur in G0 phase. But what about transcription and translation? Yes. Transcription and translation occur in G0 phase, but no replication. So now this is important right here, this line, because at this point where it goes from G1 to S, something has to happen to trigger the cell to wake it up like, hey, you need to start synthesizing DNA. What's gonna do that? Do you guys know what's gonna do that? I hope you do, because it's really important for the board. And that's gonna be your growth factors. Your growth factors are going to bind to a tyrosine kinase class of receptor. The growth factor, when it binds to this tyrosine kinase type of receptor, it will then trigger synthesis of DNA, and then thus the cell cycle continues. Now, this whole portion occurs like from G1 to S to G2. That occurs during interphase. I don't know if you guys, that arrow there is interphase. So next we're going to talk about some important components that make up DNA. And these components are referred to as nucleotides. And then we'll also talk about nucleosides. Basically the same thing without phosphate. We'll get to it. Okay, so what are nucleic acids? Okay, so nucleic acids found in the nucleus. What's found in the nucleus? DNA and RNA. That's exactly what nucleic acids are. It's just DNA and RNA. And what's DNA and RNA? Well, DNA is deoxygenated ribonucleic acid, and RNA is ribonucleic acid. So, but what makes up DNA and RNA? Well, they're composed of, they're really assembled by different combinations of nucleotides which have a specific structure. So nucleotides are basically just a 5-carbon sugar, such as pentose. Well, let's just write it out. Okay, so they're a 5-carbon sugar. Right, such as pentose. Plus a nitrogenous base, so a base, plus, before, well, I'll just, yeah. All right, plus phosphate. There could be 1 to 3 of them. Thus, tide, nucleotide, tide means 3, so a tide of phosphates. It could be 1 phosphate. AMP, for example, adenosine monophosphate. It could be adenosine diphosphate, adenosine triphosphate, ATP. So these 3 components make up a nucleotide, which a whole bunch strung together make up a nucleic acid. So what is a nucleoside? So I just said the tide was because of the phosphate. If we cut out the phosphate, that's a nucleoside. Whoa! So just this and this make up a nucleoside. Okay. So basically, a nucleoside is a simpler form, and I'll give you guys some examples up on the screen while I write this down. So now let's break down what each of those components that make a nucleotide, how they can vary. So remember, we have the bases, the phosphates and the sugars. Let's talk about the sugar first. So a 5-carbon sugar can be either there's 2 different types. So we have the molecule ribose and deoxyribose. Okay, these are the 2 types of sugars that can be used to make up a nucleoside or tide. Now, basically, ribose is going to have the hydroxyl group, the OH, and deoxyribose will not have that. So deoxygenated ribose, deoxyribose. That's it for the sugars. That's all you really need to know. The next component are the bases. Now these bases are also called nitrogenous bases. Nitrogenous, to be generated from nitrogen, nitrogenous bases. So obviously they're going to have some nitrogen in them because of that. So we have 2 different types, the purines and the pyrimidines. I'm going to pull up a picture now to show you guys the difference instead of writing out every single thing to save time. So as you guys can see, the purines, and now there's an acronym called pure as gold, you guys have heard. The A for as stands for adenine, and the G for gold stands for guanine. So pure as gold, that's just referring to A and G, adenine and guanine. These are the purines. And purines are really distinguishable because they have 2 rings. So that just, should tell you right there that it's a purine. Now, how do you distinguish between adenine and guanine at that point? Well, you need to ask yourself what's at the 12 o'clock position on the ring. So you'll notice that if it has on the 6-carbon ring, what's at 12 o'clock. So if it has an amino group at the 12 o'clock position, then you already know if it's amino, it's adenine. And if it's got an oxygen group at the 12 o'clock position, then that right there should tell you that it's guanine. So, pretty simple. Now, those are the purines. What about the pyrimidines? Now, the pyrimidines, there's 3 of them that we need to know that make up DNA and RNA. Well, so the pyrimidines, there's cytosine, uracil, and thymine, and they're all single rings. And just like we were looking at the 12 o'clock position on the ring on the purines, now on the pyrimidines, we're also going to be looking at the 12 o'clock position of the pyrimidines. So cytosine has the amine NH2 group at the 12 o'clock position. So right away, it's the only one that does, you know that that's cytosine. The next one, basically you're looking for an oxygen group at the top. But wait a minute, uracil and thymine both have your oxygen group at the 12 o'clock position. So how do you distinguish those 2? Simple, you look for the methyl group, CH3. If you find the methyl group, that should tell you right there that it's thymine. So thymine is super distinguishable because it's got the oxygen at 12 o'clock and a methyl group off to the side. Uracil, just the oxygen group on top. Cytosine, amine group on top. That's the pyrimidines. It's that simple. So you guys need to know what's in DNA and what's in RNA now. So the purines are in both DNA and RNA. And the pyrimidines depend. So if it's cytosine, it's in both DNA and RNA, off top. If it's uracil, then it's only in RNA. It's a simpler molecule, it's only in RNA. Thymine has an extra carbon, it has that methyl group in there, it's a little more complex. Thymine is only in DNA. So now if you guys see a strand of DNA, you can distinguish it from RNA with that little presence of that methyl group in there. That should tell you whether or not it's DNA or RNA. So basically, those are the bases. And the next thing is a phosphate group, right? And these phosphate groups are connected to these nucleosides to make them nucleotides. And the connection is formed by a phosphodiester bond. Now, that's where it connects on top and bottom. So it's like diester, it's two ester bonds. Ester bonds are strong, and the fact that it's phosphate, it's going to give off a negative charge. And if you got 3 phosphates on there, then it's going to be a highly polarized molecule, which is responsible for why DNA is negatively charged in the first place. And we're going to see why that's important in a second. So moving on now, here's some examples of some nucleotides that you guys may be already familiar with. So on the carbon, if you guys notice that there's these little dashes next to the numbers around the pentose molecules in a nucleotide, that just denotes their prime carbons. It's not to say these carbons are more superior, it's just to differentiate those carbons numerically from the base carbons, the carbons of the base. So just so you guys don't get confused about whether something's prime or not prime. It's just for the nomenclature. For example, now I want to see how you guys can name these, because it's pretty simple, right? So if we start out with a base, and let's start out with thymine. Let's just start with thymine. Okay, so we start out with thymine. What would the nucleoside be called if we have a nucleoside containing thymine? So thymine is the base, and thymine is only in what? DNA, right? So thymine's only in DNA. So what kind of sugar then? Because I'm asking what would the nucleoside be. So a nucleoside, remember, is a base plus a sugar. Now what would the nucleoside be? So we already know the base is going to be thymine, but the sugar is either going to be ribose or deoxyribose. Now if thymine is only in DNA, it must be deoxyribose, because that's only in DNA. So the nucleoside with thymine would be called deoxy, because of the deoxyribose thymine. See? It's pretty simple to name them. And then, now what would the nucleotide, we throw some phosphate in the mix. And it becomes a nucleotide. What would that be called? Depends. How many phosphates are you throwing in there? How about one phosphate? Deoxy. Oops. Oh wait, I have it fine. So deoxy. Deoxy thymine monophosphate. Oops. Can't even write. Phosphate. Right. Oh well. Deoxythymine monophosphate. If there's only one, if there's two phosphates, it's not mono then, it's di. So deoxythymine diphosphate would be written little d, big T, big D, big P. DTPP. I didn't write the other one. DTMP if it was monophosphate. And if it was three, then obviously tri. So D, big T, big T, big P, DTTP, deoxythymine triphosphate. So now you guys know how to name nucleosides and nucleotides based on their bases and sugars and number of phosphate. It's not really, you're not going to be asked to do that on the boards, but it's important so that you can differentiate the nomenclature, so you can read the language that may be in a question. It may not be the question, but it may be a part of the question, right? So now we got to talk about some important things to consider when talking about how the DNA strands come together and connect. So, or how they're together in the first place, basically. So the important thing to realize is that these nucleotides are connected to each other down one strand with phosphodiester bonds. But that single strand is linked to another strand by hydrogen bonds on the bases. So it's not too important as how many hydrogen bonds per base group pairing. You know, the G to C has 3 hydrogen base pairs. The A to T or the A to U, if it's RNA, has 2 hydrogen bonds. It's not too important, but what is important is that there's a 3' end on one strand and a 5' end, and that's what's going to give you polarity, right? So the sequence is always specified from 5' to 3' if they're giving you a sequence of nucleotides. So you may see something like TCGA, right, to 3'. So that's a sequence right there that you may see. Now it's always, if these aren't there, the 5 and the 3' aren't there, it's just TCGA, then it's assumed that the 5 and the 3' are there. It's only 3' to 5' if it's actually written 3' to 5'. They may do that to try and screw you up a little bit, but then you gotta remember in your head that if it was 5 to 3', then it'd actually be A, G, C, T, right? So watch out for that, because that's like, it's something simple, but they can get a lot of people by doing that. So basically, let's take a short break and we'll return in a minute. Okay guys, we're back, and now I'm going to pull up an illustration that's just showing how these nucleotide monophosphates, which assemble DNA, how they're linked together to each other by phosphodiester bonds and to other strands by the hydrogen bonds. So as we could see up at the top, we have the 5' end going down to the 3' end on one nucleotide strand, on one DNA strand. And then the other strand is going to be anti-parallel to that strand. So it's going to be going from the 3' end to the 5' end. So Now the 3' end is always attached to the hydroxyl group, the carbon that attaches to the hydroxyl group on the sugar, and the 5' end is what's going to attach to the monophosphate group. So that's a diester bond, and those two bonds together make up the diester bonds. So basically, as you guys can see, G always pairs with C, and A always pairs with T if it's DNA. If it's RNA, A is going to pair with uracil, with U. I'm just using the letters to save time, but just so you guys know, guanine is going to pair with cytosine. Adenine is always going to pair with thymine or uracil if it's RNA. And these were found by a guy named Chargaff, and those are what's called Chargaff's rules, is that the proportion of this molecule will always equal the portion of that molecule that it pairs with. There are times when this doesn't happen, and that's mispairing, and we'll get into that a little more as we go along. Okay, so next I just want to briefly mention that DNA can be denatured, and that just means that it could be split. Now, how do you disconnect the two strands of DNA? There's actually 3 different ways you can do it. And the first one is pretty simple, and that's heat. The second one is an alkaline pH of a solution, or an alkaline environment will denature DNA. Also, certain chemicals such as formamide and urea are commonly used to denature DNA. Now, to renature DNA, you cool it. So I just wanted to mention that. So now let's talk about the organization of DNA. And when talking about the organization of DNA, it's important to consider things that the fact that it's a coil, it's a double helix. Now it can either be very loosely bound or very tightly bound, and if it's very, very tightly bound, or if it's overwinded or underwinded, then basically that's caused by strain, and that's when it's referred to as supercoiling. Supercoiling just doesn't mean super tightly wound, it can also mean super loosely wound. But it's just, it's not normal. So supercoiling occurs due to strain on the molecule caused by the under or overwinding of it. So I just wanted to touch on that. And so now let's talk about what a nucleosome is. Because a nucleosome is basically a package of DNA. And a nucleosome is basically proteins that are wrapped in DNA. And these little wrapped up proteins go along a string, and a string wraps around them. And this is going to wind and make up a chromosome. But if we look a little closer, you can actually see that there's certain things, certain proteins within each nucleosome, and these proteins are referred to as histones. And these histones are positively charged, and that is what makes them want to wrap themselves in a negatively charged thing such as DNA. So why are they positively charged? Well, histones are very rich in arginine and lysine, the amino acids. That's what gives them their positive charge. And DNA, of course, is negative because of the phosphodiester bonds. So basically now there's 5 different histones that we're expected to know. And the first one is H1, and then H2A, H2B, H3, and H4. Now, every one of them except for H1 has a copy of itself within the nucleosome. So there's a copy of H2A, there's a copy of H2B, there's a copy of H3, and a copy of H4 in each nucleosome. Now, why did I not include H1? Because H1 is on the outside and it's acting as almost like a knot on a burlap sack or something. It's tightening. It's on the outside just to tightly compact all the histones within the nucleosome. It's pretty cool, and that's why it's found outside the nucleosome. It has a different structure. It's a little bit bigger than the other histones, and basically it's what's responsible for condensing the nucleosomes to form 30 nanometer strands, as opposed to just little 10 nanometer strands without the H1 histone to compact them. So without H1, these little loosely compacted 10 nanometer strands are just more loosely compacted DNA, and that loosely compacted DNA is going to be subject to nucleases, which can go and tear it apart. So it's important to have this H1 so that it can become more condensed. But then when it becomes very condensed, it becomes harder to express that gene as well. So basically, each chromosome in G0 or G1 contains one linear molecule of double-stranded DNA. So now for a second, let's talk about chromatin. And chromatin is just basically chromosome material, right? So chromatin is usually two types. You have euchromatin and heterochromatin that occurs in cells in interphase, right? So heterochromatin has much more highly condensed chromosomes that aren't really expressed, right? Because of what? Because of H1 being there. And the euchromatin is a lot loosely condensed. And let me pull up a picture to show you guys the difference. Let's see if I could find, okay, here's an electron micrograph of a nucleus that's in interphase. And as you can see, here's euchromatin, here is heterochromatin, and then as well, you could also see a nucleolus up in there. So basically, this is showing you the different density. If it's a lighter color, then that DNA is very loosely wrapped, more light can get through. If it's darker, then obviously it's more condensed and less light can get through. So now what's going on in the light stained parts? What kind of expression, what kind of gene expression is going on? Or is there any gene expression going on? The answer is that transcription is going on in the light stained areas. These genes are being transcribed because they're accessible, right? So replication will also occur in these spots, but it depends on the cell. Replication will occur to cells that are in G1 phase, not G0 phase. So if it's G0 phase and they say, well, here's a nerve cell, here's a muscle cell, there's no replication that's going to be going on in that euchromatin. Now, if it's a G1 phase, then the cell is undergoing interphase and expression is happening and transcription is occurring so that DNA can be synthesized more. So all these things are going on in euchromatin in G1 cells. What's going on in the darker portions, otherwise known as the heterochromatin parts of this picture? Well, if we look at the heterochromatin, obviously we already know that it's being more highly condensed, but basically it's showing that these genes are inactive. They're not being expressed. So inactive genes would be the answer. But what else could be in those dark spots? I'll give you a hint. This extra thing would only be there if it was a female. The answer is bar bodies. Bar bodies are basically leftover extra X chromosomes. An X chromosome is obviously relating to females, and only females have extra X chromosomes laying around in their cells' nucleuses. So this would be a bar body. Like I mentioned, genes have to be exposed for them to be expressed and for RNA and proteins to be made. This is hugely important. So for example, in a beta cell, would you expect to find insulin? Yes, the gene for insulin is obviously in the beta cell. Now, what about in the alpha cell? Yes, it's just inactive. Now, insulin is actually, the gene for insulin is in every cell of the body, believe it or not. It's just activated and being used in the beta cells. Okay guys, so just to finish up, we're going to talk about how DNA is packaged in different degrees, right? So if we were to draw it out from more active to less active, the more active DNA would be more loosely bound, right? So the most active DNA we can have is the straight up DNA double helix. Now let's add some histone in there, some H1 in there, so let's add some histones in there except H1. Let's add some H2A, let's add some H2B, some H3, some H4, and before you know it, nucleosomes are now starting to wrap around, and the double helix becomes 10 nanometer nucleosomes. So it's gotten a little thicker, but it can still be expressed. So now let's add H1. Let's add the histone H1 to the mix. So now you could see that the 10 nanometer chromatin becomes 30 nanometer chromatin, which is also referred to as nucleofilaments. So now we can even take it further and condense it more if we add a certain protein. And these proteins are called scaffolding proteins. And once they're added to that 30 nanometer thick chromatin, they become super thick fiber loops of this 30 nanometer chromatin. We can even go further and add more proteins. We don't need to worry about the names of those proteins, it's low yield, but if we go further and add more proteins, it'll condense even more to heterochromatin. And this heterochromatin is what we see in those dark spots on the slide that I showed. And the heterochromatin is very less active than the loosely packaged DNA. Because this is really tightly packaged, it's really hard to access. You can't even see the DNA. It's so tightly packaged in this heterochromatin. So basically that sums up the first lecture, guys. In summary, I just want to touch on what we talked about, okay? So what are nucleic acids? Well, DNA and RNA. And what are nucleotides? Well, nucleotides are nucleosides with a mono, di, or triphosphate attached to it. If it's DNA, it's nucleotide monophosphates linked together. And then if it's double-stranded, then those are bonded by hydrogen bonds. The nucleotide monophosphates are binded to each other with phosphodiester bonds, which gives off DNA's negative charge. And the polarity of these strands is denoted by 3' to 5', and then on the other strand, the reverse, 5' to 3'. If we're given a sequence of nucleotides for a DNA strand, we must always assume that it's 5' to 3' when we read it, unless they give us the 3' to 5' in the damn sequence. That's important. Double-stranded DNA or RNA. Now, that's bonded by the hydrogen bonds. And the sequences, of course, are anti-parallel. So, when we're talking about eukaryotic DNA, the DNA that's within us, it's packaged into nucleosomes by H1. It's further condensed by H1. The H2A, H2B, H3, and H4 are what are contained within the nucleosome. So the 10 nanometer fiber becomes 30 nanometer fiber, becomes much thicker because of the higher condensed DNA. Now we talked about euchromatin versus heterochromatin within the nucleus and how to distinguish that and what is going on in those spots. So remember, in euchromatin, genes can be expressed, so therefore RNA and proteins can be made in the euchromatin, and transcription and translation can occur. So in heterochromatin, it's too condensed to express. It's basically the DNA's waiting to be expressed, right? So guys, thanks for joining me. I'm really happy you're here for this course. That was just the first lecture. We're gonna get a lot deeper, but it was very important to set this one first because the certain nomenclature that we use is going to really make sense here coming up in the bigger picture of things. In biochemistry, I used to actually hate biochemistry, but I really began to love it once I began to see that everything is the way it is because of biochemistry. So I encourage you guys to go over the materials that I've provided and prepare yourself for the practice quiz before the next lecture, just to supplement you guys. And be sure to email me with any questions. Thanks guys, I'll see you soon. Bye.